In this lecture, I would like to discuss with you the strength and limitations of the different types of models one can use in systems biology. Let's start with statistical models. Statistical models are very useful as bookends in defining biological functions at a systems level, especially sort of at very sort of distal relationships such as a gene and a disease. But it can also be a gene and a phenotype, and the phenotype can be at a subcellular, cellular, or sort of tissue organ levels. But let me give you the example from the gene and the disease because this is most often used. And here is an example from what is a genome-wide association study where one can have a case with a certain type of disease. One can have a control. One can get say DNA or, from these patients with the disease and the control patients, compare what things have changed and make molecular correlates to the physiologic functions. And these kinds of correlations can provide context to understanding how these genes may be involved or are likely to be involved in the disease. Statistical models, like I said before in a couple of the other lectures, statistical models have the ability to capture probabilistic relationships. Not all relationships or not all edges between nodes, whether it's gene-gene interaction or protein-protein interactions, occur in a deterministic fashion, which is that they're always, when the two are always present, they occur. They may be the interactions and hence the edges are operational under a variety of conditions, and hence have a probabilistic relationship. And here is an example, and statistical models have the ability to capture these probabilistic examples, relationships. Here is an example that I took from a paper by Eric Schatz and his colleagues, where they devised a statistical test that would link a gene to a phenotype, and this can be and this linkage can be in the gene is called a locus or a position in the chromosome to a gene to a sort of a phenotype or a clinical trait called T, and there can be a variety of ways in which they can be linked using a variety of statistical tests. And they devised, in this paper, devised a statistical test that sort of is built on a assumption of causal relationships. These kinds of statistical tests, although they're not very widely used as yet, tell us that even when you don't know all the details in between, one can clearly link a gene to a phenotype and thus allow for building of this allow for an understanding of how the system might behave and have predictive capability of systems behaviors. So those are the positives with regard to statistical models. Now, like everything else, statistical models have their limitations, and one of these limitations is that statistical models obscure mechanistic details. Statistical models do not consider mechanisms. So for instance, if you are looking at a relationship between a gene and the level of messenger RNA in a statistical correlation model, one wouldn't really consider whether the change in levels is due to CNV, copy number variation, or DNA methylation of the promoters, or so on. So statistical models just correlate two nodes with respect to their levels or activities, but does not really address mechanisms. Statistical models, of course, do not consider details of pathways of action in measuring relationships between genes and phenotypes. So what happens is that when you don't consider these mechanistic details, sometimes you miss out on clues, and these missing clues, often if they're modifiers of the phenotype, can lead to sort of limited can lead to limitations in your ability to predict whether a gene or changes in a certain gene or a locus will cause a certain change in the disease, because that may be only valid under certain conditions or not under other conditions. So not considering the mechanistic details can lead the correlations to fail. And then when it fails, because you don't have a knowledge and understanding of the mechanisms, if one is not able to explain why the correlation doesn't work under a certain circumstance. So this is a limitation of statistical models. The other, of course, limitation of statistical models is that they do not provide dynamic or spatial information. Statistical models most assume a steady state relationship between two entities, say for instance the gene or the protein and the phenotype being produced. And so because of but in real life things are changing all the time. So for complex diseases, phenotypes change with respect to time and treatment, such as like if you treat somebody with a drug, a cancer patient with a drug, they eventually develop resistance to the drug. So it is often necessary to capture the dynamic details of the phenomenon in the model, and so the statistical models cannot satisfy this requirement. For many biological functions also, especially at a cellular level, spatial specification is essential for understanding function, and spatial specification cannot be represented easily in statistical models, so this is also a limitation. The next type of models one can think about are, of course, network models. And network models are essential for understanding the topology of the system. I have taken here a network diagram of the subway system of New York City to tell you that if you want to get from, say, Bronx, which is up here, to Brooklyn, that's down here, you need to sort of understand how the subway systems, or you need to know how the subway systems run through Manhattan to get from one borough to the other. Networks in some ways are similar to subway maps because they can tell you how you can get from here to there, that is from, say, genes to proteins and functions and phenotypes, and what routes can be taken, and of course what the points of connections are. So all of these are sort of important characteristics of the system, and so it is essential to understand topology or organization of the system, without which one doesn't really have an understanding of the system as a whole. The other great advantage of network models are they are flexible and can be used for multi-scale descriptions of a system. Since the user defines the edges, uh, this allows for networks to be built across a single scale or between scales of interaction. So for instance, you can build a network that is where all the connectivities are at the level of the protein-protein or protein-DNA interactions. You can also build a network where certain molecular entities are connected to certain tissue or organ characteristics. So this ability to build these networks within scales and between scales allows for one to connect key reactions at one level to functions at another level. This is actually very important, especially in terms of drug actions, because most drugs are small molecules. They interact with their targets at the protein level, but the effect of the drug is really seen at the tissue or organ or indeed the whole organismal level. So having networks, both molecular networks and networks that propagate from cells to tissues to organs to whole organisms, is sort of a very important aspect, and network models are sort of useful for doing this. Network models also provide good descriptions of regulatory or information processing capabilities of systems. Uh, as I told you in the previous lecture, directed science-specified graphs are required to identify the presence of feedback and feed-forward loops. When one identifies these kinds of motifs, one gets sort of an evaluation or understanding of the capability of information processing, and identifying these loops allows then one to draw inferences how the system as a whole will be able to process information. Information processing is knowing about information processing is important because it alters input output relationships and how the system responds to stimulus. Actually, change in input output relationships leads to change in state change or state change in many cell types, such as normal cell becoming a diseased cell, or inactive cell becoming an activated cell, as is in the case of immune cells when they see an external pathogen, and so on. So changes in cell state can most often results in physiologic changes. So to understand the changes in cell state, and consequently such as the origins of disease, one needs to have directed science-specified networks of the cells and tissues of interest. And this is sort of a major requirement towards which will sort of enhance our understanding at a systems level, and is an active area of research by many researchers in the systems biology of various cells, phenotypes, and diseases.